let's now talk about some autonomic drugs. To better understand some autonomic drugs, we'll first need to focus on the nerve terminal and the nerve synapse. Now, how are neurotransmitters such as acetylcholine made? So within the presynaptic nerve terminal, as we see here, we need to synthesize a neurotransmitter. We'll need to store it in a vesicle, we'll need to release it into the synapse, and finally, we'll need to degrade or reuptake that neurotransmitter. So all of these multiple steps are going to occur at the end of the neuron or the nerve terminal, seen here. There are going to be also some various drugs that we'll throw into the picture, and these drugs can interfere with the various steps. So let's start off by looking at the cholinergic nerve terminal. Choline is going to be taken into the neuron from the extracellular space and combined with acetyl-CoA by the enzyme CHAT, which stands for choline acetyltransferase. Now, we need to dust off the biochemistry cobwebs and remember how acetyl-CoA is formed. We're going to take our glucose, we're going to turn that via glycolysis into a pyruvate, and then that pyruvate is going to transform into the acetyl-CoA by the pyruvate dehydrogenase. So that's how we get our acetyl-CoA. We're going to have our choline, which is going to go into the cell. We're going to use our CHAT enzyme, and we're going to produce our acetylcholine. Now, we can introduce our first drug, which is going to be hemicholine, which is going to compete with choline in this step and prevent choline from being uptaken into the presynaptic neuron. So that's going to be one way we can prevent neurotransmitter synthesis of our neurotransmitter. Once the acetylcholine has been made, the next step is going to be to package it into presynaptic vesicles. Now we have a whole bunch of vesicles, but I blew one up. Here we can see the acetylcholine, which gets packaged into pre-made vesicles. And these are going to travel to the end of the nerve and wait to be released, which you can see kind of down here. Now we can introduce another drug, which is going to be visamicol. This is going to be an experimental drug that can compete with acetylcholine in this step, which prevents packaging into vesicles. Now next, we're going to have depolarization of the presynaptic neuron. So we're going to have a depolarization wave come down. It's going to depolarize the end of the nerve. Now, this depolarization causes voltage-dependent calcium channels to open, and it's going to lead to an influx of calcium. Now, this leads the vesicles to fuse with the presynaptic membrane, which ultimately is going to release acetylcholine into the presynaptic cleft. We can also talk about botulinum toxin, and this inhibits synaptobrevin, which is simply a snare protein that mediates the transfusion or the fusion of the vesicles with the presynaptic membrane. Hence, botulinum prevents acetylcholine release. And this is going to prevent the muscle contraction, and it's going to lead to a flaccid paralysis. Now, do you remember that sweating is going to be mediated by muscarinic receptors as well? So botulinum toxin actually prevents sweating as well. So once inside the synaptic space, we have our acetylcholine, and it can bind to a postsynaptic receptor, like this receptor here, to mediate its effect. Also, it can be enzymatically degraded by acetylcholinesterase, which is going to split the choline and acetate. It's going to take our acetylcholine, and it's going to convert it into two products here. And then choline, we have the choline here, it's going to be reuptaken. It's going to start our process off again. And it's going to be taken up into the neuron, and it's going to be resynthesized into the acetylcholine. Thus, we have a cycle of how we're going to make our neurotransmitter. So here's another question I have for you. What enzyme is responsible for the formation of acetylcholine? And then also, I have another question. What are its two substrates? So we just covered the formation and degradation of acetylcholine, so this should be an easy one. If you're struggling with this question, make sure you go back to the previous slide and review this pathway one more time. Now, the enzyme that we're talking about is choline acetyltransferase, which is also known as the CHAT enzyme. It's going to take that acetyl-CoA and take a choline, and it's going to convert it into our neurotransmitter acetylcholine. So I'll pose even one more question to you. What enzyme is going to be responsible for the breakdown of acetylcholine in the synaptic cleft? Do you remember what that was? That would be acetylcholinesterase. Now, its shorthand is going to be ACHE, acetylcholinesterase. So that's going to be the enzyme that is going to be responsible for breaking down the acetylcholine that is within the cleft. This is going to be a very important enzyme, and we'll spend some time later talking about this in the lecture. Now let's take a look at the noradrenergic synapse. How are the catecholamines synthesized? The noradrenergic synapse is where the catecholamines, such as dopamine and norepinephrine, that's where these are synthesized, assembled, packaged, and released. First, the amino acid tyrosine, which is going to be a precursor for all catecholamines, is going to be transported inside the nerve. Now, once inside, this tyrosine is going to be converted to dopa, and then it's going to be converted to dopamine. So a tyrosine analog, called metyrosine, can interfere with this step by competitive inhibition. Metyrosine was initially used as an antihypertensive agent, but because it non-selectively blocks all catecholamine synthesis, it's going to have a wide range of side effects, and therefore it's rarely used today for purposes other than research. Now this dopamine gets converted into norepinephrine, where it gets packaged into vesicles that can be released into the synaptic cleft, and that cleft is right there. So here, these vesicles containing norepinephrine will be the purple spheres. So reserpine is going to be an antihypertensive and antipsychotic, and it's a drug that can cause catecholamine depletion. Reserpine inhibits the packaging of catecholamines into the presynaptic vesicles. Guanethidine is an antihypertensive that replaces norepinephrine in vesicles, and it also decreases the amount of norepinephrine released from the synapse. Bertilium is an antiarrhythmic agent that also blocks norepinephrine release. So in summary, we have metyrosine, reserpine, guanethidine, and bertilium that have different mechanisms of action. However, they're all going to work to decrease the sympathetic outflow by decreasing neurotransmitter release. So now, just like in the cholinergic neuron, an action potential depolarization will cause the release of norepinephrine into the cleft. And once in the cleft, the norepinephrine acts on receptors to exert their effect. Now, some drugs actually increase the concentration of norepinephrine in the synaptic cleft. This is going to lead to increased sympathetic outflow. So have you ever wondered how cocaine works? Cocaine, tricyclic antidepressants, and amphetamines all increase the amount of norepinephrine in the synapse by preventing its reuptake into the presynaptic neuron. So here we see the catecholamines trying to be reuptaken into the neuron, and these will block that step. Amphetamine also is going to stimulate norepinephrine release into the synaptic cleft. Okay, let's talk about how does a cholinergic synapse and a noradrenergic synapse differ. So a crucial difference between the cholinergic synapse and the noradrenergic synapse is going to be based on the reuptake of the neurotransmitter. We'll start off with a brand new picture here where the norepinephrine has already been released into the synaptic cleft. Now, if you can recall from the cholinergic neuron, where acetylcholinesterase breaks down our ACH into choline and acetate within the synaptic cleft. Now, on the other hand, we're going to have MAO and COMT. And these are going to be the enzymes that are going to be responsible for breaking down our norepinephrine or our catecholamines, but these are going to be located within the neuron, not in the actual cleft space itself. Therefore, the main method for clearing norepinephrine from the synaptic cleft is going to be reuptake into the neuron. So let's introduce an alpha-2 receptor here. Do you remember what the function of activating an alpha-2 receptor does? Sympathetic stimulation of alpha-2 receptors, so we take our norepinephrine here and we stimulate it, it's going to provide negative feedback and reduce sympathetic outflow. So we're going to decrease norepinephrine release. So if we release norepinephrine into the cleft, it's going to go back onto an auto-A2 receptor, and it's going to decrease norepinephrine release, kind of acting as a negative feedback mechanism. Now, we also have activation of M2 receptors by acetylcholine. This is going to inhibit norepinephrine secretion. So we're going to decrease norepinephrine release. Whereas activation of angiotensin II receptors is going to increase norepinephrine release. So as you can tell, the body is a complex system of checks and balances. This neuron here is going to have multiple receptors, and it's going to have multiple points of regulation. So make sure you're comfortable with how neurotransmitters are synthesized, broken down, and regulated, since these topics do make great board questions.